Very good evening and welcome, dear students. I cordially invite you all to this session, 30 question discussing session. Today we discuss chapter number 6, strategies for enhancement of food production. Strategies. First question. Outbreeding is an important strategy of animal husbandry because it outbreeding. Let us come to outbreeding later. It is useful in overcoming inbreeding depression. It is useful in overcoming inbreeding depression. This is a broad one. This is a broad answer. Let us look into the detailed or precise answer to this question. For that, let us come what is meant by outbreeding. What is meant by inbreeding? What is meant by outbreeding? Inbreeding is the mating of closely related animals. Inbreeding is the mating of a closely related animal. Outbreeding is the mating of unrelated animals. Outbreeding is the mating of unrelated animals. So you differentiate between inbreeding and outbreeding. The former one is the mating of closely related animal. Other, mating of unrelated animal. Inbreeding is the strategy to improve animal breed. Outbreeding also. But when we practice or when we perform inbreeding as a strategy for improvement of animal breed, there is a drawback. There are many advantages for inbreeding. Inbreeding increases the homozygosity. It helps to develop pure line. It eliminates the inferior gene. It allows the accumulation of superior gene. Like that, many advantages are there. Inbreeding. It is mating of closely related animals of the same breed. Closely related animals of the same breed. But it has a disadvantage. The disadvantage of inbreeding is inbreeding depression. Disadvantage of inbreeding is inbreeding depression. What is inbreeding depression? That is reduction in the yield and vigor of animal. Reduction in yield and vigor of animal due to continued inbreeding. Due to continuous inbreeding, the yield of the animal, the vigor of the animal may decrease. Vigor of the animal may decrease. This decrease is called inbreeding depression. This decrease is called inbreeding depression. How to overcome this disadvantage? The disadvantage, as per the option, it can be solved or overcome by outbreeding. But when we come outbreeding, there are three types of outbreeding. But there are three types of outbreeding. That is outcrossing, crossbreeding, outcrossing, crossbreeding, and interspecific breeding. Interspecific breeding. If a question is asked, which is used among these for overcoming inbreeding? This question says it is outbreeding. This question says it is outbreeding. But there are three types of outbreeding: outcrossing, crossbreeding, interspecific breeding. So this is used for overcoming inbreeding depression, not these two. So out of the three types of outbreeding, outcrossing is used. What is outcrossing? That is mating of unrelated animal of the same breed. What is meant by unrelated animal? Unrelated animal means these animals have no common ancestor. Suppose A and B mated together. A and B has no common ancestor. For 4 or 6 generations back, up to 4 or 6 generations back, the mated animals have no common ancestry. Mated animals have no common ancestry. So, mating of unrelated animals of the same breed. Crossbreeding, mating of animals belonging to two different breeds. Mating of animals belonging to two different breeds, not the same breed. Here, interspecific breeding, mating of animals belonging to two different species. Two different species, but they are closely related. Two different species, but they are closely related species. Mating of animals belonging to two different closely related species. Among these two, outcrossing is used to overcome inbreeding depression. Broadly speaking, outbreeding. But if the option gives these one, it is outcrossing is the answer. Next, interspecific hybridization. Interspecific hybridization. Interspecific hybridization is, as we said, two different species. Two different but related species. You know, horse and mule mated together to produce horse and donkey mated together to produce mule and hinny. It is an interspecific breeding. Horse belongs to one species, donkey belongs to another species, so it is an interspecific breeding. Next, homozygous pure lines can be obtained by. Homozygous pure lines can be obtained by mating of related individuals of the same breed. Related individuals of the same breed. There is a single term to refer this process, that is inbreeding. That is inbreeding. Inbreeding is used to develop homozygous related individuals. So pure lines means homozygous. Homozygous pure lines in cattle can be obtained by. So one of the advantage of inbreeding is inbreeding increases the homozygosity. Inbreeding increases the homozygosity. Increases the homozygosity. So that is about the homozygous pure lines. Since inbreeding increases the homozygosity, it is used, inbreeding is used to develop pure lines. What are pure lines? Homozygous individuals are referred by the term pure lines. Next is a very simple question. that is domesticated birds used for food and their egg. It is called poultry. It is called poultry. Which one of the following the products of apiculture used in cosmetic and polishes? Cosmetic and polishes. Apiculture. Honeybee keeping. Honeybee keeping provides two important products. One is the primary product, honey. Second, it is the byproduct or second important product is wax, bee wax. Here, honey is used as an ingredient for Ayurvedic medicine and some other purpose. Industrial use, cosmetic and polishes, the component or the product used from the product obtained from apiculture, that is honey beekeeping, that is maintenance of honeybee cages, growing honeybee. It is bee wax, not honey. Honey, it is not in cosmetics and polishes. Next, which of the following is not a marine fish? Katla is not a marine fish. It is a freshwater fish. All the other three are marine fishes. They are marine fishes. Katla is a freshwater fish that is in pisciculture or in fisheries it is used. Then, next question asks about marine fishes. What are edible marine fishes? Edible marine fishes, hilsa and pomfret. Answer is C. Both A and B, they are marine fishes. Question number 8. When breeding is between different breeds, breeding is between different breeds, it is called crossbreeding. It is called crossbreeding. What is outcrossing? Breeding or mating between unrelated animals of the same breed. Unrelated animals of the same breed. Interspecific hybridization. Hybridization between two different species. What is inbreeding? Breeding between or mating between related animals of the same breed. Related animals of the same breed. Related animals of the same breed, inbreeding. Unrelated animals of the same breed, outcrossing. Breeding between two breeds, crossbreeding. And interspecific breeding between. So these three together, it is called outbreeding. These three together are called outbreeding. Breeding between unrelated animals. Next. A tricky question, here it is given. The process which result in the identification of superior animals and superior females of the same breed. A trick is played in this question. And if the trick works, we will be in trap and commit a wrong answer. We select a wrong option. What is the trick played? Two tricks are played. One, selection of superior male and female. Selection of superior male and female. We study in the strategies. We study in a breeding program. In all breeding program, superior male and females are selected and they are mated together. They are mated in pairs. So in all breeding program, there is selection of superior male and superior female. So all we can say. But here the second trick played is same breed. The selection is from the same breed. If the selection is from the same breed, it may be either inbreeding or outcrossing. Selection is from the same breed. It is either inbreeding. Inbreeding is the mating of related animals of the same breed. Outcrossing, mating of unrelated animals of the same breed. Unrelated. So, same breed, animals from same breed are mated either in inbreeding or in outcrossing. So, the answer is either inbreeding or outcrossing. So, answer is inbreeding. No outcrossing is here. Crossbreeding between two different breeds. You see, it's not same breed. Interspecific breeding between two species. There is no question. Two different species never belong to same breed. Two different species never belong to same breed. Never belong to same breed. So that is about this question. The trick played, identification of superior male and female, is the first step in all breeding program. In all types of breeding, whether it is inbreeding, whether it is outbreeding, whether it is outcrossing, crossbreeding, interspecific hybridization, in all methods, selection of superior male and female is done. It is done. So we may be puzzled. However, we come to the answer. But the same breed, it will help us to select the answer. Breeding of animals belonging to same breed, it may be either inbreeding or outcrossing. In all others, it may be between two breeds or between two species. Okay, also right. Now the answer comes to question number 10. Inbreeding is carried out in animal because it increases homozygosity. One of the advantage of inbreeding, mating of related animals of the same breed, it increases homozygosity. What are the advantages of inbreeding? It increases homozygosity. It eliminates the inferior genes because we will select only the superior animals. It will eliminate the inbreeding. It will eliminate the inferior gene. It gives a chance to accumulate the superior genes. So, disadvantage is inbreeding depression. Okay. Next. When breeding is between unrelated animals, note unrelated animals, including individuals of the same breed. We can stop here, but we should completely read it. Unrelated animal of the same breed. Unrelated animals. Unrelatedness is explained in the question in the later part. That means 4 to 6 generation, they have no common ancestor. 4 to 6 generations back, they have 4 to 6 generations back, there is no common ancestor for the mated animals. So unrelated animals belonging to the same breed. Unrelated animals belonging to the same breed. The later part of the question explains or narrate how the unrelatedness explains. They have no common ancestor 4 to 6 generations back. So simply the question can be converged to mating the animals of the same, mating unrelated animals of the same breed. It is actually outcrossing. It is actually outcrossing, but there is no outcrossing here. But outbreeding, one of the outcrossing, 3 methods we studied, that is outcrossing, crossbreeding, and inter-specific breeding. Okay. So outcrossing is the answer, but it includes in outbreeding. So we select outbreeding. Next. Which of the following is considered to be the root of breeding program? Root means fundamental basis of plant breeding program. The fundamental basis of plant breeding program lies in genetic variability. Genetic variability. If there is no genetic variability in a species, take it granted, hypothetical. If there is no genetic variability, in a species, we could not conduct plant breeding program. We could not improve that species through conventional breeding program. If there is no variation, if there is no genetic variability, then only, if there is genetic variability, then only there is a question of plant breeding program. So genetic variability is the fundamental basis. It is the root of plant breeding program. Question number 13. A variety is given. Sonaliga and Kalyan Sona. Sonaliga and Kalyan Sona, they are varieties of wheat. What is the advantage? What is the achievement in these varieties? They are high yielding and disease resistant. They are developed by plant breeding program. Through plant breeding program, they are high yielding and disease resistant. Then next it is semi-dwarf varieties of rice. How they are developed? Semi-dwarf varieties of life means small life. Height is not that much. Not too tall, not too small. They are semi-dwarf varieties. How they are produced? What is the advantage of semi-dwarf? The tall varieties. If the varieties are tall, they will lodge down. They will fall down due to the weight of the grain. Due to the weight of the grain, they will fall down. The disadvantage, when the rice plant fall down and the grains are on the floor, on the soil, it will germinate. The semi-dwarf varieties are advantages. They will not fall down. How the semi-dwarf varieties were produced? It is by crossing IR8 and Taichung native 1. Taichung native 1. This is a variety of rice. This is another variety of rice. By crossing these two varieties, semi-dwarf semi-dwarf varieties were produced. Semi-dwarf varieties were produced by hybridizing IR8 and TN1. Okay. IR8 is a variety developed in, it is in the next question, so we can go to it. Improved rice variety. IR8 has been introduced in India from. It is introduced in India from Philippines. Why? IR8, it was developed in International Rice Research Institute. International Rice Research Institute was located in Philippines, in Manila, the capital of Philippines. This rice research station is situated or located. So IR8, a high yielding variety of rice developed in International Rice Research Institute, and they were introduced to India from there. Okay. IRRI, International Rice Research Institute. The next question is the International Rice Research Institute, IRRI. It was located in Manila, of Philippines. Next, in maize, presence of high aspartic acid, low nitrogen and sugar content protect them from. This is a physiological characteristic of maize plant. This physiological characteristic protect the maize plant from stem borer. That is the insect which will bore the stem, making some holes, destroy the plant. There are many morphological and physiological characters which do not prefer by the insect. So these morphological and physiological characters provide insect resistance. Insect resistance. Okay. So hairy leaves, it is an insect resistant character. Smooth leaves, it is an insect resistant character. It is an insect resistant character. These are morphological characters. Some physiological characters also give the ability to resist the insect. So here it is the physiological character of maize. Then 18. Lysine and tryptophan are essential amino acids. Essential amino acid. What are essential amino acids? Those amino acids cannot be synthesized in our body. They are called essential amino acid. That is, amino acids which cannot be synthesized in our body, which are not synthesized in our body, it is called essential amino acid. Essential amino acid. Okay? Then, but there are certain amino acids which are synthesized in our body. Which are synthesized in our body. It is called non-essential amino acid. Why they are called non-essential? Because these amino acids are synthesized in our body, we need not require external source. We need not require external source. But in essential amino acid, in the case of essential amino acid, we require an external source to get these amino acids, mainly plant source or animal source. The food from plant source, the food from animal source, we depend. So lysine and tryptophan, they are essential amino acids. To get these essential amino acids, we depend upon certain source. But plants can synthesize all the 20 amino acids. Plants can synthesize all the 20 amino acids. But we could not synthesize all the 20. Some we could synthesize, but some others we could not synthesize. So they are called essential amino acids. They could not synthesize. Question number 19, okay. Himagiri developed from hybridization and selection for disease resistance against a pest population. Himagiri is a variety of wheat. It is a variety of wheat. It is a variety of wheat. It is produced variety of wheat. This variety can resistance against stripe rust, hill bunt, etc. So there are some other varieties. Brassica, the variety Pusa swarning, it is resistant to white rust. Cauliflower, Pusa subra, Pusa snowball, it is resistant to black rot, curl, blight, black rot. Cowpea, it is variety Pusa common, bacterial blight. Chili, Pusa sadabagar, it is chili mosaic virus, tobacco mosaic virus, and leaf curl. So Pusa, you can see the prefix Pusa in many cultivated varieties of many cultivated crop. What this Pusa indicate? Pusa is the name of a place near Delhi where the headquarters of Indian Agricultural Research Institute, Indian Agricultural Research Institute is located. This place is called Pusa. So the varieties produced from Indian Agricultural Research Institute, its headquarters is situated in Pusa near Delhi. So the varieties produced from this research station, it is known by the name, the prefix name Pusa. So that is the reason behind adding the prefix Pusa. Okay, next. It is question number 20. So in an organized breeding program, what is meant by collection of variability? Collection of variability means germplasm collection. That is, before the beginning of a breeding program, in which crop plant breeding program is planned, the first thing should be done is germplasm collection. That is collection of all the varieties of that crop. Collection of all the varieties of that crop. For what purpose it is done? collection of all the varieties in the form of seed, in the form of plant, in the form of any other material. The germplasm is collected. Collection of all the varieties of a particular crop, it is called germplasm collection. So it should be done. The primary thing should be done in a plant breeding program is collection of germplasm or germplasm collection. It is the collection of all varieties of crop plants in which the breeding program is planned. But for what purpose? This is to study and understand about the variations existing in the crop. Germplasm collection is meant for the study of variation existing in the plant crop. What is the importance of studying variation? We mentioned in an earlier question, variation, genetic variation is the basis of all breeding program. After having a thorough knowledge about variation existing in a food crop, then only we could conduct a breeding experiment in a successful way. So to study the variation, germplasm should be collected. Next. Answer. Next question. In mung bean, resistance to dash and dash were introduced by. We studied an example of this plant, a variety of mung bean developed by mutation breeding. It is resistant to yellow mosaic virus and also powdery mildew, and it is developed by mutation breeding. These two diseases, the plant is resistant. What is mutation breeding? That is breeding programs incorporated with a mutation. Here mutation is used. What type of mutation? Induced mutation is used. What is induced mutation? Artificial mutation. How the mutation is induced? Using mutagens. What are mutagens? That is agents capable of causing mutations. Agents capable of causing mutation. They are mutagens. Mutagens may be chemical mutagen or physical mutagen. The most commonly used chemical mutagen is EMS. That is ethyl methyl sulfate. Ethyl methyl sulfonate. It is a mutagen. The physical mutagen is gamma rays or gamma radiation. These two are the two important mutagens, commonly used mutagens used in mutation breeding. So a plant breeding program in which induced mutation, artificially created mutation is used, it is called mutation breeding. The basis of mutation breeding, in mutation breeding, plants are subjected to induced mutation using suitable mutagens. Then, after induced mutation, the plant is evaluated or observed whether any favorable, whether any desirable characters occur due to mutation. If a desirable character occurred due to mutation, such a character or such a plant is exploited in mutation breeding. Question number 22. another example is asked. That is, resistance to dash in Abelmouscus esculentes was transferred from wild species and resulted in a new variety. The new variety, it is called Parbani Kranti. The resistance is yellow resistance against yellow mosaic, isn't it? Yellow mosaic virus. Abelmouscus esculentes, bindi, or okra, are commonly called lady's finger. So, lady's finger, it is susceptible to the cultivated variety of Abelmouscus esculentes. It is susceptible to yellow mosaic virus. The cultivated varieties, they give yield, but they are not resistant to yellow mosaic virus. They are not resistant. There is no preventive method for a viral disease also. So, the diseases occur in Abelmouscus. This variety was made, or Abelmouscus esculentes, it was made resistant to yellow mosaic virus by introducing a gene from wild variety of Abelmouscus. Wild variety of Abelmouscus. Probably you may think that white is not used in cultivation. Wild variety has disease resistance power, ability to resistance disease against yellow mosaic virus, ability to resist the disease against yellow mosaic virus. Then white is not used in cultivation. It is not used in cultivation because the yield is poor. It is not economical to cultivate this wild variety. The yield is poor. Maybe sometimes the taste of the fruit which is used as a vegetable may not be excellent or may not be favorable. But it is disease resistant. By making use of genetic engineering, biotechnology, tissue culture, the modern tools, molecular tools, the disease resistance gene in the wild variety had been identified, and this gene is taken out and it is introduced into cultivated variety of Abelmouscus with the aim of making the cultivated variety disease resistant. Making the cultivated variety disease resistant. So, high yielding quality is there. Now, disease resistance property is introduced. It is a genetical mechanism. It is introduced to Abelmouscus by transferring the gene from a wild variety. Wild variety. So, disease resistance is a character controlled by the genes, not a chance character, not a character acquired by luck. It is a character present due to the genetic composition of the plant, genetic constitution of the plant. So, it is introduced, making the plant transgenic plant. Transgenic plant. Answer is E. So, Abelmouscus esculentes, variety Parbani Kranti, a new variety. Then, select 23, no 22 we select. Select biofortified crops from the following. Biofortified crops. So some are wrongly matched. Some are wrongly matched. Answer is E. All except 3 and 5 are correct. 3 is wrong. We could not accept it. Because in paddy, it is biofortified for iron, not copper. It is paddy biofortified for iron. Why we cannot accept? So bitter gourd and mustard, it is biofortified for vitamin C. Biofortified for vitamin C. All others can be accepted. All others can be accepted. Maize hybrid, it is a biofortified variety of maize. Biofortification, they contain high lysine and tryptophan content. They are rich in lysine and tryptophan. Wheat, the variety name is Atlas 66. It contains high protein. Carrot and spinach, it contains vitamin A. Spinach and bathua, it contains iron and calcium. What is biofortification? That is production of crop varieties having increased nutritional quality. Production of or development of crop varieties having increased nutritional quality. The nutritional quality may be increase in protein, amino acid, mineral, vitamins. Protein, amino acid, minerals, and vitamin. 24, ask about single cell protein. Single cell proteins are produced by culturing microorganisms. Single cell protein is a mixed type of protein. It is a type of protein obtained from culturing of microorganisms. A cheap method of producing protein. It is in laboratory using waste materials. Microorganisms are cultured to produce protein. Single cell protein, they are good protein. They are rich protein, good protein, but low fat content. Which organisms are used here and which organism it is used? Here all are correct. Answer is E, all are correct. Spirulina, it is a blue-green algae used in single cell protein. Candida utilis and Saccharomyces lipolytica, they are fungi used in single cell protein production. Methylophilus methylotropus is a bacteria used in single cell protein production. So different types of organisms are used to produce single cell protein. Blue-green algae, spirulina, fungi, candida and Saccharomyces. Then bacteria, Methylophilus methylotropus. Okay. Question number 25. Plants produced by tissue culture will be genetically identical to the original plant from which they are grown. Identical plants. Plants having morphological and genetical similarity. They are known as clones. So but vegetative propagation we had seen what are clones. But plants produced from tissue culture also, the progeny is genetically similar, the progeny is morphologically similar, so they are called soma clones. Answer is B, soma clones. Clone Also genetically and morphologically similar, but it is used in vivo vegetative propagation, normal vegetative propagation. Tissue culture is in vitro vegetative propagation. Tissue culture is an in vitro vegetative propagation. The normal vegetative propagation by planting a stem cutting, by planting some other propagule, it is called in vivo. 26 match the following. The answer is C. Answer is C. So an extended table is given. Methylophilus. We know methylophilus. It is used for single cell protein. Methylophilus. It is used for single cell protein. Then fortified, it is used for vitamin C. Then Pusa swarning, resistant to wheat rust. Pusa swarning, wheat rust. It is a variety of brassica. Then D, it can be matched with a 5. Transgenic plant we had seen Parbani Krati. Then Pusa savani, it is in the next question I had given it. Pusa savani. It is a variety of binti shoot and football. Okay, Pusa savani. E, it is 2. Resistant to fruit and shoot borer. Resistant to shoot fruit borer. Pusa savani. Next, the last one, Pusa kamal. Pusa kamal can be matched with a 3. Resistant to bacterial blight. Resistant to bacterial blight. It is in cowpea. So answer is C. 27 also, I match the following. Answer is D. One can be matched with U. Because Kalyan Sona is a, yes, this is a new information. This is Kalyan Sonalika. So every new variety, dear students, every new variety in the research station, it has a number. It has an ID number, what we said very specifically today. The ID number, the identification number of Pus Sonalica, it is HD 1553, not in NCRT, I think. HD 1553. Then crucifer can be matched with P, because crucifers, it is black root of bacteria. Then here, last page it is there. In cauliflower, it is okay, crucifer, black root. So paddy semi-dwarf variety, Taichung active 1, because this was a parent in developing semi-dwarf variety. Cowpea, it is Pusa kamal. Cowpea in the previous table, cowpea Pusa kamal, bacterial blight. Pusa kamal is a variety of a cowpea. Then R can be, that 6 can be matched with R. Flat bean Pusa sem 2. It is resistant to jacids, insect resistant. It is resistant to jacid, flat bean. Pusa sem 2, resistant to jacids. So that is, answer is D. Next, 28. It is, answer is A. Resistance to powdery mildew in monk bean is by mutation breeding. Resistant to yellow mosaic virus in Abelmoschus esculentus, transgenesis production of transgenic plants, then resistance to tobacco mosaic virus by hybridization. Answer is A, 29. What is protoplast? Protoplast is the total living content. Total living content. So what should be removed? Cell wall should be removed. Why? Cell wall is a non-living one. Cell wall is non-living one. So plant cell without cell wall is protoplast. Protoplast is the total living content of the cell. Protoplasm is the liquid part, not protoplast. Protoplasm with a membrane is called protoplast. Protoplasm with a cell membrane, protoplasm with a plasma membrane is called protoplast. So protoplast is the total living content of the cell. Total living content. 30. Listen. How to isolate protoplast? How to isolate protoplast? To isolate protoplast, we have to remove cell wall. We have to remove cell wall. To isolate protoplast, we have to remove cell wall. So, to remove cell wall, two main enzymes, pectinases and cellulases are used. The main components of cell wall. Cellulase digests cellulose, pectinase digests pectin. They are the two main components of the cell wall. By using this enzyme, the cell wall can be removed and protoplast can be isolated. So here we wind up this session.